Good morning. Did that wake you up? Great morning to be in Washington, D.C. How's everyone doing? I can't hear you. All right. So my name is Chris Bersalis, and I'm the chairman of the Public Policy Committee of the National Board of Trustees. And thank you for joining us this morning for the opening of the conference portion of this week's program. So I hope everyone had a successful day on the Hill yesterday and that you were able to get some well-deserved rest last night. So I'm pleased to say that my chapter from Western Pennsylvania had a great day. We received some very good feedback from members of Congress and their staffs, and we helped build support for the fight against this disease. And while not every member we met with was completely embraced our issues, they did embrace our cause, which is important. I can unequivocally say that we made a difference and that our presence was not forgotten when we left those rooms. That's the power of our message, and it's a message that we must continue to deliver loud and clear, not just here in Washington, but when we return to our hometown communities. The meetings that you all attended yesterday are incredibly important. You put a face on this disease, and as Jane Gilbert mentioned in her opening remarks on Wednesday, you make this disease personal for members of Congress. They see it firsthand in all of you. You know, I'm new as the chair of the Public Policy Committee and relatively new to the Board of Trustees. And when we met with some of the more senior members of Congress and some of the more senior staffers, I asked them, what can we do as an organization to really step up our game in advocacy and be more effective? And just about to every single one of them, had the same comments. They said what you're doing on the Hill is extremely, extremely important, and you're very effective. You're probably one of the more effective groups. But they all pretty much said the same thing. That Advocacy Day is just one day. You know, there are 364 days in the year which we must also advocate. And you know, they said you have to take this message to your communities. It's not just what we do here in Washington that makes a difference, but it's what we do throughout the year that really makes a difference and has the most impact. So it's whether you're attending a town hall meeting in your state, scheduling a meeting with your congressman's district office, inviting members to walk or support group meetings, or simply sending an email or making a phone call to Washington, all of these things matter. All of these things make a difference. And all of you are the reasons that we will succeed. Now, I'd like to take some time this morning and recognize some of our chapters who really exemplify the view that advocacy is a year-round activity. These are chapters who go the extra mile to make sure our concerns are on the minds of their elected officials throughout the year, just as we live with ALS throughout the year. The efforts of these chapters have had a real impact on our fight. The first Advocacy Achievement Award is presented to the Kentucky chapter. So, under the leadership of Board President Sean Mullinex, Public Policy Chair Connie Houston, and Executive Director Mary Bacon, the Kentucky chapter has grown to be one of the most effective chapters for public policy. The chapter has embraced public policy as a way to accomplish its mission. They recruit new advocates to our cause. They encourage people to sign letters to Congress at their walks. They distribute letters and updates to those in their service area, and they continually look for opportunities to advance our priority issues and expand their grassroots ability to make a difference in Washington. They take action when it's needed and routinely will make personal calls to Hill offices. Congratulations to the Kentucky chapter. Now, I know the Kentucky chapter has their walk this weekend, so I believe Sean has returned to Kentucky today, but I'd like to invite Kentucky advocates Joe and Frances Wise to come up and accept an award on behalf of the chapter. Joe and Frances. Thank you very much for all you do and your chapter does. The next Advocacy Achievement Award goes to the Rocky Mountain chapter. Public Policy Chair Josh Rudin has done a remarkable job ensuring the chapter is fully engaged with its congressional delegation. Not only those in Colorado, where the chapter is based, but also in Wyoming, the other state served by the chapter. Josh makes the trip to Washington each year and also regularly meets with members of Congress and their staff when they're away from Washington back home in their state. Through these and other efforts, Josh and the chapter staff, led by Executive Director Pam Rush-Negri, have generated support for ALS issues among their congressional delegation and advanced our cause. Thank you, Josh and the Rocky Mountain chapter, for all you do. Come on up. Great job. The third and final Advocacy Achievement Award goes to the Greater New York chapter. Led by Chapter Executive Director Doreen Gordon, the chapter quite simply does it all year in, year out. It's no coincidence that the two House members who champion funding for the ALS Registry, Representatives Elliot Engel and Peter King, are both from the chapter's service area in New York. In fact, the New York congressional delegation is among the strongest supporters of our cause among any state. This is because the chapter volunteers and staff like Daryl Cochran and Doreen have made public policy and advocacy a priority in everything they do, whether it's having members of Congress participate in chapter events, recruiting new advocates who live in key congressional districts, or collecting advocacy letters at all their walks, including one of the nation's largest walks on Long Island. Congratulations and thank you to the New York chapter for doing so much to support people with ALS in New York and across this great nation. Come on up. Come on up, guys. Congratulations, guys. I love New York. Great job, guys. So, while we just recognized 3 chapters for their efforts and achievements in advocacy, I want to thank all of our chapters and everyone here with us today in this room and online for your efforts to advocate throughout the year. Give yourselves a well-deserved round of applause. So next, I would like to present the Rasmussen Advocate of the Year Award. The Rasmussen Advocate of the Year Award is presented to the individual or individuals whose personal initiative and efforts increase awareness of ALS and the ALS Association through education and advocacy at the local, state, and federal level. Now, this award is named in honor of Dean Rasmussen, a former national trustee who helped to build what is now the Public Policy Department, and whose vision has helped to make advocacy a key component of our mission to find a treatment and a cure. Now, this year's recipient is a researcher, a physician, a professor, an advocate. In the ALS world, this guy's a rock star. He has an MD, a PhD, a master's in clinical research science. He's a tenured associate professor of neurology at Duke, director of the Duke ALS Clinic, and chief of neurology at the Durham Veterans Affairs Medical Center. He's published more than 60, in his spare time, he's published more than 60 ALS articles and is the outgoing chairman of the North American ALS Research Group, the ALSRG. He has won countless awards, including the American Academy of Neurology's Patients Advocate of the Year Award. He also founded ALS Untangled, which helps us to sort through the myths and facts about alternative and off-label treatments for ALS. And in fact, he'll be presenting twice during this conference today, so a doubleheader, including during the FDA session and the breakout session on the myths and facts about ALS. So to us, he is the consummate advocate. Year after year, he participates in this conference as part of the Jim Catfish Hunter chapter delegation. He attends a fly-in event that is held in February to meet with congressional appropriators, and perhaps most significant is that he encourages people to speak out and become advocates every single chance he gets. In fact, last year he recruited nearly 50 advocates to make the trip from North Carolina to Washington for this conference. He understands the power of advocacy. You can see this in everything he does. He's an outstanding advocate, and he certainly knows how to make a fashion statement. This dude is a stud. The recipient of the 2013 Rasmussen Advocate of the Year Award is Dr. Richard Bedlack. You are the man, Dr. Bedlack. That's awesome. Before we begin the research session, we'd like to play a greeting from Senator Lisa Murkowski of Alaska. As many of you know, ALS has struck Senator Murkowski's family. Pat Dwyer, the husband of Senator Murkowski's cousin Jenny, is currently living with ALS, so our fight is also her fight. She's one of the strongest champions in the Senate, helping to increase ALS research funding through her seat on the Appropriations Committee and working to build support for ALS issues among her colleagues. Senator Murkowski usually attends this conference in person and really wanted to be here today, but she was unable to participate this year because she is attending her son's graduation. However, she did record this message for all of you.
Good morning, this is Senator Lisa Murkowski. I'd like to thank Steve Gibson, the ALS Association, all those that have been involved in putting together this week's events. I do apologize that I can't be with you in person. I'm out with our oldest son as he graduates from college. But if that were not the case, know that I would be with you. It's always an honor to be part of the annual gathering in Washington to meet with other ALS families, just like mine, who are struggling with this disease and want to work together to improve where we stand now and hopefully one day find a cure for ALS. I want to thank each of you for your advocacy on Capitol Hill. I know you were out there yesterday. I know that you're going to be out there in your communities, but what you have been doing to educate lawmakers by really bringing a name and a face to one of the most horrific diseases that affects nearly 6,000 Americans every year. I just thank you so much for your advocacy. Now, I am pleased to be able to report some amount of good news this year. This past year brought some real progress in the fight against ALS. In addition to an unprecedented full day of ALS testimony from patients and advocates, this was heard by top doctors and scientists at FDA. There were also some very important policy changes that were made to the 2012 FDA Safety and Innovation Law. It now requires the Secretary to facilitate the development and expedite the review of drugs that are designated as a fast-track product. These are those drugs that are intended for the treatment of a serious or life-threatening disease or condition. Now, the FDA law also now requires the Secretary to expedite the development and review of breakthrough therapy drug that provides preliminary clinical evidence indicating that the drug demonstrates substantial improvement over the existing therapies. These funding programs and the federal policies could yield significant results for orphan diseases such as ALS, and they could also set a path forward for private dollars to be invested in ALS research, an area that, of course, we know is critical. Now, another achievement that we saw in 2012 was that the Department of Veterans Affairs concluded that ALS is a 100% service-connected disability, regardless of the progress of the disease. This allows veterans to automatically qualify for health and disability benefits, regardless of what stage of the disease that these folks might be in. These are all significant victories, and I thank you. I thank you for what you have helped to do to bring these victories, small as they may seem to some, but bringing them forward. Now then, looking out onto the horizon, I'm told that the Packard Center for ALS Research at Johns Hopkins has made substantial progress in identifying the most common cause of ALS. This is the gene mutation. Researchers are working to build some new therapies that may be able to treat a major subset of ALS with this mutation. It's this type of forward-looking approach by the collaborative teams at Johns Hopkins that will enable pharmaceutical and biotech companies, as well as the academic clinical community, to someday find effective disease-altering therapy for ALS. Now, I'm also optimistic about the initiative that President Obama unveiled in early April. This is the BRAIN initiative, which stands for Brain Research Through Advancing Innovative Neurotechnologies. I think we all know that we need to know and understand more about the brain. The BRAIN initiative proposes $150 million for NIH, the DARPA, and NSF. Strong interest has also been expressed by private foundations, including the Howard Hughes Medical Institute and the Allen Institute for Brain Science. This initiative, I believe, gives hope to patients that are battling diseases who are looking for the answers, just as we are for ALS. I'm committed to working with Dr. Collins at the NIH and with the President to understand better the brain and the diseases which we recognize we don't fully understand and grasp. Now, I will leave you with this. If you ever consider yourself alone, just look to some of our ALS heroes who've been here this week, who've given their time to advocate for ALS. You're all part of a club that knows all too well what some 30,000 Americans are struggling with every day. It is through your determination, your care, your advocacy, and what you all do collectively that we can and we will make a difference. And one day, we will find that cure for ALS. Thanks so much for all you do.
Thank you, Senator Murkowski. Let's give her another round. So we have a great program today. In a few minutes, we'll hear from Dr. Lucie Bruijn, the association's chief scientist, who will share how our Treat ALS program is advancing the fight to develop treatments. We'll hear from the FDA about how the agency works and the role they're playing in the ALS drug development approval process. And at lunch, we'll hear about ALS clinical trials. We'll have some great breakout sessions this afternoon as well, and I look forward to participating in them with all of you. Throughout the day, I encourage you to visit the exhibitors in the ALSA Marketplace. And remember, if you have your ALSA Marketplace passport stamped at every booth, you'll enter a drawing to receive a free iPad. We'll hold that drawing for the iPad during the closing ceremony today at 4:30. So thank you all again for being here this morning. Now I'd like to turn things over to our CEO, Jane Gilbert, who will introduce the morning's first session.
I'm going to be very brief this morning, but I do want to take this opportunity to extend a special thank you. With this many people in the room, it's not often that we have the opportunity to really express our appreciation for what you do as a collective group. And certainly this last couple of days has been, as I said this morning, exhausting, I know, but exhilarating as well. And the thank you that I want to extend this morning goes to a select group of association chapters who have gone above and beyond the call of duty to help us with our research efforts. These are chapters who have either by their board decision made a donation to our research efforts, or who have encouraged donors, and some of you sit among us this morning, who have restricted gifts to our research. And so the following chapters, I would like you to hold your applause, but should be acknowledged and thanked for their tremendous support of our research program. The Alabama chapter, the California chapters through the California Tax Checkoff Campaign, the Central and Southern Ohio chapter, the DC, Maryland, Virginia chapter, the Greater New York chapter, the Greater Philadelphia chapter, the Iowa chapter, the Keith Worthington chapter, the Minnesota, North Dakota, South Dakota chapter, the Orange County chapter, the Oregon Southwest Washington chapter, the St. Louis Regional chapter, the Texas chapter, and the Wisconsin chapter. Thank you so much. They have given, as I said, specific gifts to help our research program as we move forward. And certainly I want you all to join me in welcoming Dr. Lucie Bruijn to the podium, who is going to kick off this morning with our Treat ALS program. Thank you so much.
Welcome everyone this morning to the research session, and thank you again on my behalf for all that you do to make sure that research remains a very prominent program as part of the ALS Association. Thanks to Public Policy and their hard work for a fabulous opportunity this week to be on the Hill and to really advocate for more funds for ALS research. Steve particularly gave me this opportunity to really also highlight ALS's research program, and I will, when I have the opportunity, indicate how this aligns with all the efforts going on in the government. So one of the things I really wanted to emphasize is that although there are fabulous opportunities through the DoD and the CDC and NIH, the ALS research program and Treat ALS does have unique research opportunities that would not happen without this kind of funding in the association. One of the important things to note, and it was discussed yesterday at the town hall, the program is truly global. And in fact, NIH really doesn't fund international research. I've sat on council, and there are from time to time some foreign applications, but they go through hoops and through a lot of rigor to be sure that they are the ones that should be funded as opposed to the US studies. I think the importance of that is that we all appreciate we don't know where the answers will come from, and it really doesn't matter. It just matters that we find the therapies and the treatments and the cure. And so actually, as a team together globally, we can do much more effectively. What we can therefore build are very large global consortia in genetics and biomarkers. And in fact, the achievements we have made to date is really part of international consortia. And I will talk a lot about the royal we, I will talk a lot about our programs. Today is really, in my presentation, a focus on the ALS Association's research program and the impact that we've made. We often support risky research, and this is also very critical. The NIH, the CDC, the DoD requires very rigorous peer review, which is appropriate. But often it takes a little bit of risk-taking to get the preliminary data, which will then get the significant dollars from CDC and from DoD or NIH. We are flexible. We fund any time of the year. We do have certain cycles, but if a good idea comes to the table, if I hear of something exciting, I try and make it work at that very time. We also facilitate and manage partnerships, and this is really critical because a project on its own without the support will not go anywhere. It needs constant encouragement, it needs the network that we can bring other people to the table with expertise. So a lot of my time is spent in really building that network to make it effective. What's very exciting, and I'll highlight this further in my presentation, is the public-private partnerships, our effort to make partners with the academic sector as well as the industry sector to really get things into the clinic. And we do often I hear from my colleagues how wonderful it was to get your starter grant, your initiating seed funds, because now I can get the big money from the NIH, and that happens to many, many of our investigators. And most importantly, I think we really see the future of ALS researchers through our Milton Safinovitz postdoctoral fellowship program. And in fact, I'm so excited that we had this year 10 fellows that we would like to fund. We only have sufficient money for 5 fellows, but the research is out there and we need more and more support. And just to highlight some research successes, for me today, I wish I could say to you the biggest success is there's a treatment and a cure for ALS. And so there are different ways to measure success. I can't tell you that today, but I think it's important to measure our successes as we go along that path. And I think we will ultimately, I'm absolutely confident we will get there, but we have to also take the steps on the way. The discovery of new genes is in large part thanks to the investigators that we fund, the unique partners and consortia that we have generated to very rapidly support the new gene discovery. I'll talk about C9. Through building resources, we have built rat models, we have built stem cell models, we have built biomarker opportunities. We really bring researchers to our programs because it's only if you have the tools will you ever be able to do the science. And I like to tell the story that when I started in 1993 as a scientist in Don Cleveland's laboratory, we would maybe gather a group of 100 people at most for any kind of discussion or meeting. And now it is thousands and thousands of scientists worldwide who are committed to figure out this very challenging problem and finding treatments for the disease. We were also the first organization in ALS to embrace translational research, and that was really the time that I began the TREAT programs to bring good ideas from the lab through academic and industry partnerships to the bench. There are many organizations, and NIH now, through NCATS, has a significant translational program, but none as focused and dedicated to ALS research as we do. We also were the first disease to use antisense oligonucleotide and bring it to the clinic, and this is extremely exciting. It would not have happened without this opportunity of a public-private partnership. We also have the first publication for induced pluripotent stem cells in ALS research, also very critical. I forgot that I had a roaming mic. I thought I had to stand here, so sorry. I was hugging it this way. And also, what was very important was a surgical development that was required for the stem cell trial that Neuralstem is doing. We funded Nick Boulis, at the time a very young researcher working in Eva Feldman's group, to do gene therapy, and it was through his creativity and his insight that he developed all the surgical technology, first on small animal models, and then adapted it to be able to now do the trials that are going to be done both for Neuralstem and for new upcoming trials. And we have increased the number of clinical trials dramatically through a wonderful partnership with the Northeast ALS Consortium, enabling to support their infrastructure. We have, through that program, also been able to build the Clinical Learning Institute, which is something that Dr. Bedlak has been very pivotal in driving. And I'm not sure if she's here today, but we hope to see her certainly at the clinical trial session. Devon Punch is our contact and liaison for all the clinical trials questions. So that's been a tremendous support for our translational programs. And then just to reinforce that it's the young scientists, the new and upcoming generations of ALS scientists that are going to be the leaders of ALS in the future. And so we are so excited to have really tremendous, very, very well published, outstanding scientists that come into our programs, often from other disease areas, to further their research work and remain committed to ALS. But it wouldn't be correct to say that we do this all alone. It really is a partnership, and it's also a funding partnership. So we support research at the NIH, at NIA. I see Dr. Traynor is in the background. We also support the Induced Pluripotent Stem Cell Consortium. We work with the Packard Center very closely now, in particular to expedite work in the new gene mutation, and we're able to very quickly fast-track some studies. Very much after the day or two after the discovery was made, we started to work with teams to help support animal model development. We work with a Therapy Alliance. For those who are not familiar with them, when you go to CVS and you put money there, that money goes to ALS research. It goes to an organization called Therapy Alliance, who we work very closely with, especially on large initiatives, such as the Jackson Laboratory Mouse Repository that we partner with many other chapters on. We work on with the M&DA UK, a European group based in the UK, also funding ALS research. We are beginning to work with the frontal-temporal dementia groups, and I'll tell you why that is. And we also work with the Alzheimer's group in the UK because of the connection with frontal-temporal dementia. And then most recently, we have a partnership with the Muscular Dystrophy Association in the diaphragm pacing trial. So it really is a teamwork, and I wanted to echo what Jane said in recognizing the chapters. I really value the chapter participation, both for your funding and also for the fact that you often bring important and exciting research to my attention, and so it is a partnership to make sure we are all aware of the activities going on in our local communities. So Treat ALS is a program that encompasses many phases of the drug discovery process, from very early discovery all the way through supporting clinical management, testing out ideas within our clinical centers, and hoping that whatever comes through that pipeline will eventually be the therapies for tomorrow. But most importantly, rather than seeing it as a linear process, it's really a back and forth. And central to all of this is those of you living with ALS. We couldn't do this research without you, without your samples, without the DNA that we get from you, without your participation in clinical trials. So any of you feeling frustrated that progress is slow, or you wish you could do more, or you can't be in a clinical trial, you can certainly be in a clinical study. And so please make us aware of your interest to participate. There are biomarker studies, there are studies to collect DNA samples. There's always a way that your participation will be highly valued. And I'm not going to belabor the point, but just to say there are many different funding mechanisms and programs that cover, as I said, from the basic research all the way to supporting drug studies and small pilot clinical trials. Currently, our portfolio is worth $15.5 million, although that will change exactly today, because tonight we will discuss and will put forward several new proposals. We have about 20 to 25 that I hope we can fund in all different categories, and this will raise the portfolio value to well over 17, 18 million, assuming that they get approved. So the key focus areas in research, and I know I've probably got a lot more I want to tell you today than I can really do, so I'm just really giving you highlights. When I talk of any of this research, it really is research that is funded through those that come to us with their applications or through processes of discussion with me. So these are all activities in our programs. Genetics, I think, is the most important. Dr. Traynor standing there will probably agree, and a lot of the work has come from his stellar group. Disease modeling is very critical for therapy development. Biomarkers, I'll highlight, is critical. Stem cell opportunities, I'll try and talk a little bit about that. Neuroinflammation, I don't have time to talk about, but it will be very interesting to see more and more drug approaches to maybe attacking neuroinflammation, and outstanding programs in looking at the dynamics of the motor neurons and how they connect with the muscle. And then, most importantly as well, RNA biology, and I'll tell you why. But I wanted to highlight, because many have asked, and I know that we have Biogen's representation here today. We will hear more about clinical trials later. Huge disappointment that the clinical trial for dexpramipexole failed. Not through lack of commitment. 900 subjects enrolled by Biogen very, very rapidly. 80 million investment. I hope I have those figures right, but at least that's the ballpark. It was based on very promising phase 2 clinical data, and we think the mechanism was the mitochondria, the energy houses of the cell. But this alerted us to the challenge that, in some ways the trial has ended and we're not absolutely sure that the drug hit the target, and that really it's the target that isn't important in the disease, or just the drug that needs to be enhanced, modified, or changed. And for me, those lessons learned, which are not necessarily so novel, we all think the similar things, has really given me a sense of how we want to move forward in terms of strategic direction. We need to play a very critical role together with partners. We won't be the only organization doing this, but really to critically validate targets for drug development. And I'll keep talking about the concept of a target and make it clearer to you. Where possible, we really need to have better biomarkers. We need to know after a trial that the trial has failed because the drug did get to where it was. It did work on the process that we thought was important, and we now no longer think that process is so important. And again, a very, very critical thing that will tell us how to move forward. And we also need to work with our clinical colleagues to see if we can improve clinical trial design so that we can be more sure of phase 2 data before we go into phase 3. And in fact, we are holding a meeting just next week to have some of those discussions with FDA representation. So it's great to put strategic goals, but can we actually meet them, and what do we plan to do? One is, I think we can validate targets better. We need to increase our understanding of the disease process and use clever tools to test our ideas. We need to enhance our academic and industry partners and work closely with contract organizations who have real skills in drug development so that we really enhance the process rather than working in our own independent silos. That has been happening, but I think more of it can be done. We need to enhance our biomarker efforts, and I think this will come through larger strategic initiatives. We have several ongoing, and there are some very focused projects that we are thinking about. And again, as I mentioned, really thinking carefully with our clinical colleagues how to design the trials. But none of this can be done without increased research funding, not only by the government, but for our internal programs. And we really need to put more funding into our research programs internally, because that will have an enormous ricochet effect on the government programs. More and more people will be able to apply, and there'll be more and more focus of ALS in the government area. So what I'm going to try and do today in the time that I have is to give you an overview quickly of the biology. I know you've heard me speak about it, but without understanding the biology, there is no target and we can't treat the disease, and I thought it would be good to run through that. I wanted to talk a little bit about drug discovery, because I think the frustration that why don't we have treatments, why is it so difficult, why does it take so long, and why does it cost so much? I wanted to give you a sense that this is a very challenging process, and those in the industry, I don't need to be told that, but it's not an easy one. And then a few highlights from our program. And I like to, again, look at this visually, and I think rather than pointing, I'll just describe it because I won't be in both sides. But really, this is a disease of the upper and the lower motor neurons. So, in fact, it's a very complicated architecture. When we think of Alzheimer's or other diseases where there's a focal lesion, that's a little bit easier to think about in terms of treatment. And I put this out especially for stem cells, because if there is such a diffuse activity of disease, where exactly is it that those stem cells are necessarily going to do the best job? And so this is what it is. You have cells in the brain, cells in the spinal cord, all with exquisite connections that ultimately have to connect with the muscle to cause their activity and cause movement, swallowing, speech, all the things that we do. And in the time that I started in ALS in 1993, a lot of it was about the motor neuron, a strong motor neuron focus. These are motor neurons either from normal or ALS patients. And what was in them was abnormal accumulations. And at that time, I focused a lot on neurofilaments. We'll talk a little bit more about that. But the field has really expanded, and we don't think about just the motor neurons. We think about its whole surrounding, the microglia that interact with it, the astrocytes, the connection with the muscle, and even now the oligodendrocytes, a cell type that we previously didn't think was involved, and Jeff and his team are doing a lot of work on. What are the causes of ALS? Well, certainly genetics. There's a strong genetic component, at least in the 5 to 10% of ALS. And I almost feel a little intimidated with having the best geneticists in the room here. But the reality is that I do think that genetics plays a big role in sporadic as well, but likely multiple genes affecting it. But all of this is interacting with the environment and certainly with aging. And I've taken a few of the things out that were question marks, because actually our list of question marks is longer. Smoking is certainly known to be associated. The military has a twofold increase, but we don't know what that factor is. But it's really the exquisite combination of all these things that cause the disease. And this, I think, is also incredibly impressive when one says, well, what are our successes over time? In 1993, we had the first mutation for ALS, and that was a very dramatic achievement. It really shifted the field phenomenally. But actually, what happened then over time, and in fact there was quite some gap, is that we now have an enormous cluster of genes that we have identified, all linked with the disease. And Bruijn Traynor promises me that in very short time, we will have all of familial ALS. So I hold you to that. I haven't given you a timeline, Bruijn. So Dr. Amit El-Chalabi is funded by us, and he actually has a database that keeps track of all these genetic changes, all the clinical information that goes with it. And this is really an enriched database which is significantly used. And of course, at the time it started and was funded on the basis for SOD1, he now has a wealth of genetic information in this. And this is this exciting advance in this pie chart that, in fact, that gap there we hope will be filled very quickly in the near future. Probably not by this big breakthrough gene, and so we see there the yellow chunk, which is absolutely amazing. This is the C9 mutation, and Dr. Traynor and Rosa Rademakers were the key people to actually find these discoveries. Very exciting that we had supported, in part, this research. I like this to give you a sense, and I'm not going to probably explain it as beautifully as Dr. Alma Al-Chalabi does, but people always ask me, so how does it really work? Why is it that I get ALS and someone else doesn't? And so I just like the concept that there is a threshold effect. There's a point at which you get the disease and a point at which you don't. You obviously have a genetic makeup that, unfortunately, some, especially carrying the dominant genes, have a very high propensity to get to that threshold. And then in combination with other things like the environment, that might even expedite that push over the threshold. So there are many factors that can burden you towards having the disease. And genetics obviously plays a very big role. What those environmental factors are together with the genetics are still a challenge to identify. But it gives you a sense, in fact, why, in fact, some people with the SOD mutation don't get the disease for a very, very long time, and for others it's very severe. They probably have modifier genes that are affecting it to cause it to be a slower progression. Challenging when it's so complex, the disease, because every disease, every person has a different trajectory, starts differently, and that makes it a very interesting but challenging disease to treat. I'm going to quickly run over these other genes, only to highlight that what was so interesting when these gene mutations were found is that they have very similar behavior. They all are involved in RNA processing, FUS, C9, many others as well. And just to give you a little bit of a sense, and what was interesting is that these genes, all you have to take away from that, are structurally quite different, quite similar. Where you see those color boxes, that's the same. These are two different genes. And so what is coming up is that we have a whole family of gene mutations in familial ALS that seem to be involved in similar processes. And I just highlight this because the actual process is so complicated. In terms, this is a nice animation that I have borrowed that really shows, if I can get it to work, what is going on in every single cell of our body. This is what's so amazing. It's happening everywhere. This is the process where messages are taken from your RNA to transcribe them correctly and to get all the messages packed correctly. This is happening all the time in every single cell in your body, and these are the genes that are involved in that process. TDP43, FUS, perhaps C9, we don't know enough yet about its mechanism. This is again showing you, once you've got it transcribed, you then have to translate it into proteins, the machinery in your cells that are going to be the building blocks for everything in your cell. Again, this is happening in every single cell in your body. And why is it then that these mutations, TDP43, FUS, perhaps C9, are actually doing something to very particular cells in the body that is making it process incorrectly and building the building blocks incorrectly? So that's why a lot of our work is focused on looking at the partners, things that are interacting that should not be interacting when there's a mutation in that gene. What is also intriguing, and I think this is a field that has been around for a long, long time, we have understood that ALS can be associated with a frontotemporal dementia. But I think that the number of people affected and having this combination was not always so clear, and it has been the genetics that has come so strongly to show us that many of the genes involved in ALS are also involved in frontotemporal dementia. Again, very interesting how two very different systems can come together with the same genetic change. And now we see ALS much more as a spectrum of diseases where, this is borrowed from Virginia Lee, on the one side you have pure ALS, on the other side you have pure frontotemporal dementia, and somewhere in between you may have a spectrum of both. This has become particularly interesting for the C9 mutation. What does genetics do for us? Well, first of all, it helps us build model systems. And from these model systems, we can test ideas and also test therapies. And we have a variety of models. Beautiful work recently published from a colleague at Northwestern University really showed some of the first models where they are mapping the upper motor neurons using very clever technology. Most of the mouse models have been looking at the lower motor neurons. They are easier to find in the spinal cord. And this is a tracking system now that will look carefully at the upper motor neurons. What is also interesting, and work by a veterinarian showed, in fact, that there are dogs that have the SOD1 mutation. And we have been supporting her work and will continue to support it because actually we think this might be a very useful tool, both for dog owners to help treat these dogs, but also to overcome the challenge where many say, well, the mouse is so distinctly far away from humans before going into clinical trials. We are actually working with these SOD dogs, and we will test some of the ideas of therapies, antisense approaches, and vaccination approaches in these dogs to treat the SOD disease. So it will be very interesting to see how this evolves. So just briefly to touch a bit on drug discovery and what translational research really means. So initially what you need to do to even start the drug discovery process is have a target, and I've talked about that many, many times. A target is a way to go in and say, if we get a drug that works on this thing, a mechanism or a molecule, we can actually affect the progression of disease. So that is the starting point of something that is translational. It then has to go through a whole process of chemistry, design. Even antisense oligonucleotides we'll talk about. They're not really small molecules in the sense that you take them once, twice, 3 times a day in a pill, but they also require a lot of chemistry. And that is typically done not so much in the academic sector, but in the industry or biotech sector. The next phase is to actually test those chemical compounds in animal models and cell model systems, and that often is done in academia or in industry. And then finally, it goes into clinical trials once all the appropriate FDA approval has been done. And who funds the clinical trials? Again, most likely either the government or the industry, because now we're running into very, very expensive research. But this is a cyclical process because clearly things, when they fail in clinical trials, will go back to the drawing board to try and identify better targets or redefine that target. Something that has happened over time and has actually put a very positive spin on it for us is that the industry really is challenged with increasing costs in research and development and really a plateau where we see that jagged line. That is a measure of the number of new entities that go and get approved for experimental trials in clinical trial by the FDA. They're not FDA-approved compounds. They still have to go into clinical trials. And that has stayed fairly level with an unusual spike at one point. But the costs have gone rocketing high. In fact, a more recent publication showed that the true cost of drug discovery and getting a drug approved is about $1.8 billion for that drug, partly because so many fail, of course. And so what has this caused us? What has happened? What has changed? Well, basically, many of the companies are now working through either contract organizations, academic partners, or most importantly, organizations like ourselves, because they see that the value of the partnership will de-risk it, reduce the cost, and actually have a much better focus on that particular disease. We are the experts in the disease. We have the people living with the disease, and we have the places to get the materials and the networks. And that's why there are so many more exciting public-private partnerships. And this just shows a little bit, again, of that drug discovery process. It starts in academia in most cases. It's the great discovery, the great idea, the light bulb idea. And it has to go through a very systematic process to get into clinical trials. Some of that is done in academia. Some of it is done in startup companies that come out of the academic sector. And a lot of it is done in the biotech sector. But where we can work as NGOs, non-government organizations, we can really broker that and make sure that those partnerships, instead of working in parallel or singly, work together as a team. And I think that that's going to be much more the direction that we go, where there are much more hubs and spokes and groups that are working in together. And I think that we play that pivotal role. This is just a quick listing of some of the companies I can think of that are working actively in ALS. Both Bristol Myers Squibb and Pfizer don't have ALS programs, but they're extremely interested in doing so. And I've had many discussions, and I believe that in time they will too have programs in ALS. But the other companies all have active programs, either with academic partners or on their own in ALS research. And this is our challenge. We have now a lot more information. We have scientists working on so many things, but we have a very complicated picture. Every single one of those boxes is a target for the disease. But which is the good target? Which is the one that works? Which is a successful one? And I think this is where biomarkers come into it. So again, highlighting the fact that the Biogen trial failed. Did the drug get into the CNS? Did it go where it was supposed to go? Did it do the biology it was supposed to do? We have all those questions remaining. We need to get in earlier to treat the disease. We need improved diagnosis. We need to know who is going to be in the trial. Are there populations that might respond better than others? How do we stratify the population? How do we improve our clinical trial outcome measures? Some fantastic work was done with Biogen and changing the measures to have both a survival and a functional score combined. We need to do more work in that kind of direction. And we need to be sure that we have pharmacodynamic markers, that we know that the drug has engaged with the target. We have currently for clinical trials ALS Functional Rating Scale, which measures gross function. We have survival. We have the combined assessment now, which is going to be more likely adopted in trials, forced vital capacity, and measurements of muscle strength. But we don't really have biomarkers that we use in trials, as in a blood biomarker or a CSF marker, to see some changes. Diagnosis, I don't have to tell you what it takes to go through diagnosis. Many of you shared those stories yesterday on the Hill. No definitive test, ruling out one thing for another, time taken, takes too long before we get to being able to treat the disease. But there are some very exciting things in the area, and this has really been significantly funded through our programs, looking for small molecules, something in the blood that might tell us that it's changing in an ALS case. And this was work done by Dave Borschelt and Jerry Shaw, who looked first of all in the mouse model and showed that neurofilaments, which are integral parts of the neuron, they form part of that long extension between the cell body and the muscle, and they're very important for the architecture. I think I briefly mentioned that that's where my research work started, because they were the blobs that you see in motor neurons at end stage. And in fact, in the mouse model, they are starting to increase and increase. It's likely telling us that it's a measure of dying cells and that they somehow accumulate. What was interesting, however, that this is not only relevant for the mouse model, but we looked in human samples and found that similar change. And in fact, when we look in several patients over time, although it is flat over time, once it's high, it stays more or less at that level. It's certainly, if you look, the left-hand panel is the ALS patients, the right-hand the controls, there's a distinct difference, and this could really be a significant marker. In fact, in the last few months, there was a publication from Kevin Boylan and his group that showed when neurofilaments are extremely high, the progression of the disease is faster. Now, that was done in a small select group of patients. We need to find out in more patients, but that could be a very interesting way to stratify our groups. And if you have a measure to say, gosh, the neurofilaments are high, those are maybe the group you would define as a group in a clinical trial, separate from those that have lower levels. So these are the beginnings of thinking about how we can do this. We have lovely work by Suad Rutkov, who's been looking at different ways of measuring muscle strength, and this is the EIM work that's been done, and really less invasive and more manual to be able to take it around. So very exciting. It's being used in the NeuralStem trial as well. This is just to tell you how it works, and in principle, it's really, once you stimulate the muscle, you're looking for a response, and this is a healthy response, and then you get the stimulation back from a muscle that's not functioning, and you can clearly see the difference. It's not as strong a response. MUNI is something that Jeremy Schiffner has worked on for years, and in fact, both MUNI and EIM muscle measures show good reliability from one center to the other, and that is, of course, critical to do in clinical trials. You need to be consistent, and we rely on combining trials in many, many centers. Other biomarkers that are being developed by Michael Benneter and others is looking at imaging technology. This is in the spinal cord. Of course, these are the first actually studies that have been done in spinal cord for ALS, and that's where the disease manifests. He both looks at the structural changes and the chemical changes with this technology. This is a slide borrowed from Martin Turner, who's doing beautiful work in the UK in his biomark study, where he's looking at brain imaging, and he shows, interestingly, if you look carefully, these are images in live time. The pictures on the left that I just showed you are a moment in time at end stage. He can show the same changes in a living patient, so you don't have to wait to end stage. You can look at early changes, and these are all technologies that have really evolved in the last few years. Again, highlighting work from his lab. One of the initiatives that I'm very excited about, and it tells an interesting tale actually, is the development of ligands, PET ligands. And EAT2 is actually the glutamate transporter that's very important on astrocytes, removing excessive glutamate. Not so long ago, we had unfortunately the failed clinical trial of ceftriaxone. Ceftriaxone was a trial to see if you could enhance the removal of glutamate or the activation of this transporter in patients, and it was actually a very interesting program that identified that ceftriaxone might help. But just the same story as DEX, when ceftriaxone failed, we also don't know whether actually did that drug affect the glutamate transport. We couldn't measure it. The timing's not exquisite, and this is what often happens in science, but this particular marker that we've developed and supported that is now being shown in a rat model and will go into human trials within the next year, could have been used to measure that in the trial. And it's just showing you how, where I think our priorities are to try and align these better, to help fund these initiatives that will be the markers for future trials. I'm going to skip over that because I have an interest of time. So this is an initiative, again, that I've mentioned to you that really shows the opportunities through partnerships with the industry, and also the fact that in ALS, we can't rely on one small molecule to do the job. I think we've got to be much more creative. It's a complicated, complex disease in which I think multiple approaches will be needed. And so through innovative approaches of antisense oligonucleotides, stem cell therapy, and gene therapy, we certainly are making advances in the fields, and I'll talk a little bit about IPS. This is the technique where antisense oligonucleotides quite simply block the production of an unwanted protein, and where we spent years trying to figure out what SOD1, the first mutation found, was doing wrong, and we still dispute, discuss, and publish a lot on that. We still don't know the exact method, and we don't know how to treat that method. What we do know now is how to block its production, and this was a fantastic and unique opportunity with ISIS to do that. This is how it is administered. It goes through a needle and through a catheter into the intraspinal space and gets directly delivered. We showed in our primate studies that you can actually get it to the motor neurons, and it then went into clinical trials and was published recently as being successful from the perspective that it was safe. It was only a safety study, so of course we don't know if it was effective to slow the disease. It was also in a very small number of patients. Because of this partnership, this company now works in Huntington's, SMA, myotonic dystrophy. All of those are moving quickly to clinical trials. There's an SMA trial at the moment. Huntington's has partnered with Roche and ISIS to move this quickly into clinical trials. Why I say that is that often ALS can be actually the leader in providing technology and opportunities for many diseases. The other reality is that as science goes along, technology improves, and so we've now got better chemistries for the antisense oligonucleotides. And a decision had to be made at this point. Do we carry on with the lesser oligonucleotide into clinical trials, phase 2 and 3, very expensive, or do we go back to the drawing board and very quickly get the better and best technologies into clinical trials? And so for those of you who feel that the field of SOD antisense is dead, be assured that within the next year, we may well be able to announce a clinical trial with better molecules. So this is Bruijn's story and Rosa Rademacher's story. And so really, it's the excitement around the discovery of C9, and also the reality that you can never be sure that one group will find it or one group alone will find it, because we funded 3 major initiatives in this area. Dr. Traynor, one of them, Dr. Rosa Rademacher, another. And the biggest consortium, the most expensive one, they felt very sad that they did not find it. They used the best and latest technology. We had the human genome colleagues, Eric Lander and his team, making back clones, but that technology was never going to find it because it was advanced to such an extent that it was chopping up the bit that we really needed to have as one large piece. And it was because the scientists and clinicians went back to the older technologies that they were able to find that enormous expansion. We are pleased that the back group did continue, and in fact we funded them because they have generated what is now required to make the model systems. So all 3 teams together have made significant contributions, and these are the publications. Very simply put, and those who are going to attend the FEL session tomorrow will hear from Dr. Traynor in much more detail. All I want you to take away from this is that that red box there in the images there is an enormous repeat, sometimes thousands in size, that should not be there. And in fact, what that repeat exactly does, we will still be debating about. We certainly know that it causes clumping and aggregates in the nucleus. And in fact, this has also been shown now when you take fibroblasts from patients, you see those same images, and this is just to hone in on that, those same clumping. So it has been reproduced. It's also seen in spinal motor neurons in that same way. And this is from ISIS. They immediately started to work, amongst other companies as well, to develop that same technology to take the principle, well, we don't want the things there, let's block it, and let's use that antisense technology to stop the production. They have demonstrated so far that they can effectively target that expansion, and they learned that in part from myotonic dystrophy, because myotonic dystrophy also has a big expansion like that. They know that if you lower the expression of this big expansion and the normal transcripts, the mouse model or the rodent model that's being tested in seems to be doing fine. So technically, we think it could be used as a therapy. And currently, they are now screening for the best one to take to clinical trials. So this is moving incredibly fast. Where I said to you in 1993, we identified the SOD1 gene, and our first publication of a clinical trial was just this year. I think that that field is going to shift much faster for C9, and that's how we've learned from our earlier investments. I already mentioned that Nick Boulis was a surgeon who developed all the technology behind injecting of spinal cells, and I'm going to move a little faster because I know I want to have time for questions. I wanted to emphasize this because often when I go to chapters, they feel the only hope is in stem cells. And I think that that is maybe misguided, and partly because the hype and the excitement that comes in the media. Stem cells are certainly an optimistic hope, but I've just told you about the antisense technologies for C9 and for SOD1. There are certainly going to be other small molecules that will be tried in the clinic. I think it's going to be a combination of things. So we cannot put all our hopes into stem cells. And in fact, stem cells at the moment are probably the most challenging. So the transplant that is being done at Neuralstem, I'm very excited. It's progressed from being in a mouse model system and getting into humans. It's certainly been shown to be safe. It's incredibly invasive, and I don't have the movie to show, but you really have to open up, and it goes right into the spinal cord, and it's really not a trivial surgery. But I think it's exciting because it really has now, in fact, it's got approval to go to the next phase and is moving forward again. So we continue to support this. We are supporting now Eva Feldman to be able to extend her patients into this study, and it's something that we're very excited about. We have, in fact, Dr. Svensson was someone who I was very excited to get into the ALS sphere because he spent most of his work in Parkinson's disease and got very excited to work in the disease when he actually met two of the board of trustees and knew of their sons that they had lost at a very young age and was quite moved by that and felt that he could really do something with his Parkinson's knowledge, and he certainly has. He has moved the idea of stem cell therapeutics forward in a slightly different way to Neuralstem. He has decided to use the stem cells as a trafficking vehicle and package into them something that the neurons like, which is in this case GDNF. So they are delivering a better environment to the dying motor neurons and astrocytes. And they are so far, they have got to the point where they've developed this. They have shown that in cell systems it works. They, in fact, have presented to the FDA, actually prior to Neuralstem, but then they realized there were some challenges with the cells that they were injecting and had to reproduce them, and that took some time. But they are now in a position where they are ready to file for an FDA approval and hope, if all goes well, to go into clinical trials next year. And so we continue to support this effort, a slightly different approach, which is very important. He was excited about the opportunities and the way it was moving forward. He did express the challenges, which is something we will work with FDA. A lot of the data from the Neuralstem trial is not public data. And so each of these individual stem cell trials have to go through a lot of toxicity and studies that could really benefit from shared data from the other trials. And so this is an area that I think we can really work with to see how we can improve that. But the other area that stem cells are really important for and very exciting for is the induced pluripotent stem cells. And this is how you take a skin cell from patients that have maybe unknown genetics and generate them into motor neurons. And I think a lot of advances are going to be made with this technology and tools. We support at NIH a consortium to develop these lines for investigators. We also work with Steve Finkbeiner at the Gladstone, who's created very creative technology to be able to look in live time. And this is just to show you that if you can look from time to time in a dish at what is going on, and then perhaps treat with a drug in that dish and see what changes, you could do some very interesting experiments in having cells that come directly from humans rather than those that are have to be modified to carry the genetic mutation, make it an even more relevant and living system. And finally, to say that we also feel strongly about our support in the NEALS network to help with the clinical trials and to make sure that centers are receiving standardized and are able to share data and make sure that all the trial data from one site to the other is relevant. And this really was through a phase 2 trial that was run through this network, was part of the motivation for Biogen to move forward to the dexpramipexole phase 3 trial. So although we didn't fund the studies to get dex to the point, we did fund actually very early studies of Jim Bennett to prove that it might be interesting. We really helped through supporting this network to help in efficiencies of clinical trial design. And it's also to give the plug that today Dr. Merit Sukovitz will be here to talk about clinical trials. And she really has done a tremendous work with her colleagues in being out there, upfront, testing all the different things, improving trial design, and will be there to answer any of your questions about specific trials. I also encourage you to join us monthly on our monthly webinars. I know I've had to go through a huge amount of information, and there's so much more to tell you. And in fact, what I do is get investigators to tell the work that they are doing and then enable you to have a Q&A with them. And it's often a very helpful way to get immediate information about what's going on in the field. We also have Devin Punch, who works with the Clinical Trials Network, who will certainly... Are you here in the audience, Devin? Oh, hi, Devin. Could you stand? Sorry to put you on the spot. She is our liaison. Thank you, Devin. When you go to the information site at alstrialspartners.org or you phone that number, she is at the other end of it. And she might not know all the answers, but she'll certainly go and find them out for you. And so she is your liaison to help you in a very complicated field of which trial am I eligible, when is it enrolling, how do I find it. Yes, the NIH has a very comprehensive site, but for all the diseases. And so what we have done, and she is working really hard at, is to make sure that the information is very accessible to you. So on our website, we have the trial site link where you can search and find information, and she is pivotal in that program. So I think with that, I really wanted to thank you all for your attention. I know it's really quite a vast amount of information. I hope I've covered some of the topics that are of interest to you, and I do welcome any questions. We certainly have some time before our break today at 11. Thank you. Questions?
Hi, my name is Bernadette, and I'm from Portchester, New York. I have a quick question. You mentioned something about the dogs with ALS. I work as a director of a specialty animal hospital, and our neurologists do see ALS from time to time. And I'm just wondering if there's any way that we can be of any help to reach out to the animal world with all the neurologists and everything that you are working on.
Oh, that's fantastic. Do you know Joan Coates? Do you work with her?
I don't know
in any
Joan,
way?
no. Jason Berg, Richard, Joseph,
I would love to. So let's connect afterwards. I'll give you
Okay.
my card, and I would be very pleased to connect you in with that, because as we think about the trials in the dogs, we would probably need to extend maybe to more communities, and I would be very pleased to help link you in with all of that.
Okay, thank you.
Thank you. Yes, there's a question here. I'm sorry, the lights are so bright I can't really see very well. There's one here at this table, please.
That was a great summary of the state of the art. And my question is, to what do you attribute the success in accelerating research? And what are the biggest obstacles that need to be overcome to keep up that?
It's That's a lovely question, and I'll repeat it. So what do we attribute the successes to, and what are our challenges? So one is, I think, the big success is that once you make a small finding, you get more people engaged in the field, and then you get more and more findings. I think it's really thanks to the commitment from the investigators, the growing pool of investigators, the opportunities and the tools, what we can bring as a community to help and facilitate that. So I think the growth has been because the science is there. People are attracted to problems they can solve, and I think there are many problems we can solve in the disease. We haven't quite got there to the cure and treatment, but we've got a long, long way to building the tools. So I think that's partly the testament to success. Technology, certainly, because with the whole gene technology changing, that's made things move faster. Communications makes things move faster. We really are a global network, because if I don't know the person, I'll soon find out about that person through a network. And so that's been amazing. It really is a global effort, and it's grown exponentially. The challenge is twofold. I think as simple as that. Funding is always the challenge. And I really emphasize again that although there is increasing money and there's a push for money in the government, it's funding in organizations like ourselves that make an enormous difference, because we can do a lot of seed funding, creative management of programs, bringing teams together in ways that really can't be done in one single institution. So I think we have a lot of flexibility, but money is always required. The biggest challenge is the disease itself. It's not an easy one to crack. I don't think it's an impossible one to crack. I'm confident we're going to get there, but it does mean that we have to be extremely creative. We have to diagnose earlier. We have to find markers. We have to understand that one disease is not the same as another disease. So the disease is the biggest challenge. But we won't be defeated by that. We just need to figure it out. And because we have exponential people working on it from all kinds of places and ideas, we will figure it out. But it's fitting it all together and making sure we keep communicating. We keep sharing our data. We keep working in teams so that when we don't find something, we figure out why. It's the interesting C9 story. In fact, I don't know if I quoted right, but whether it was 7 or 8 years, it felt like my whole entire career with the association, we were worrying about the C9. And we didn't find it, and I knew all the groups working, and they promised that they would tell me as soon as it was found. We kept confidentialities between the groups. We were all doing different things. We just couldn't find it. And it was a small technology thing. I know I dumb it down, but in principle, that was what it was. We were looking under the light, and we were there, and we were in the right place, but we just didn't know how to do it. And so it's teams of people. But I think the challenges are not unsurmountable. I think with funding and with commitment and with all of you just keeping at it and being hopeful, we will get there.
My name is Leanne Kozak. I'm with the Greater Sacramento chapter. I am a huge supporter of paying it forward. In fact, I am in my 4th clinical trial right now. But I also would like to know what specifically is being done for those of us with ALS today. And I am thinking specifically of things like compassionate use, earlier access to investigational drugs, that sort of thing.
So I'm not the best person to address that. I'm going to give you my opinion and my answer, but we will have a whole session on clinical trials later where that's a good question to re-ask again. What are we doing for patients now? Well, I didn't focus on it because I really wanted to show you the research activity. But I also think we care a lot about people living with the disease now, and symptom management is critically important and actually part of our strategy as well, clinical management programs. We have a trial going on for Nudexta. For those that have trouble swallowing and speaking, we think Nudexta may work, and so that would be tremendous to help people now with that, and it will be quite a quick trial, so once we find the information, it will be accessible to everyone. We are working with cytokinetics to test a small molecule for muscle strength to improve daily muscle strength. So these are all activities absolutely thinking about improving living with the disease while we also look for treatments for extending life. The question about compassionate use is such a complicated one. The only thing, I said it yesterday to some who were in the room, remember that a clinical trial is an experiment, okay? Just like I did lab experiments in the basic research arena and tested things and designed it in the best way, it was a test. So if you're going into clinical trials, it's a test. the answer might not yet be the answer, especially if you're in the early part of that experimental design. So if something comes out in phase 2, and maybe DEX is not the best example, but minocycline, very promising, went to phase 3. Actually, only at phase 3 did we find out it was toxic. So I will have speakers like Jesse Cederbaum, who will do a much better job of it, who is passionate that really you've got to be careful with just taking any treatment. And it seems bizarre, and many of you might come back and say to me, but look, it can't be worse for us. Look at how we've declined. I can be a guinea pig, and many people say that to me. But actually, if your position got worse suddenly in such a way that it could compromise you terribly, that's everyone's personal choice, and I can't shout out for everyone's personal choice. But I think we have a responsibility to do the right experiment. So that's what we struggle with, the desperate need to do things fast, and we're working and we're talking about that, and the desperate need to do it right. Because if we don't do it right and we think something's working and 5 years later we realize, boy, we didn't do that right and we've wasted our time and people are taking it but it's not helping, and actually we could have done a better trial and in 5 years' time those people could be helped properly, we're in that dilemma. We really want to help people now, but we want to assure that what we do is the best thing for the future. I hope that addresses some of it.
You talked about consortia and cooperation across agencies, but you didn't mention FDA. Okay. Can you...
Oh, that was, so again, it's always tricky when you're listing things. I hope all the chapters were correctly acknowledged because everyone is a special chapter, and the same thing with funding agencies. Even reading through it, I remember there were people I should have acknowledged. So FDA is an important part of this, and obviously clearly from the hearing that many were engaged with. In fact, next week the FDA will be at our meeting with a small subset of clinical trial experts to talk about clinical trial design. We are constantly engaging, and in fact, in reality, so I mentioned the FDA with Clive Svensson's work. Many of these, the antisense technologies, we come in really early and we encourage that, and we're doing more and more of that, to speak to the FDA at the beginning of trial designs, at the beginning of drug discovery, because we need to know early, they need to understand early, what are the experiments that we need to do, because we don't want to waste time. We want to educate them to say what's most important. We want them to educate us what is important for safety. So we work closely with the FDA. And in fact, even at the hearing, that was the one thing that was very positive, that scientists and clinicians accept that the FDA works very well together with us on those issues. There are areas we can all work better and faster, but as such, there's a very good working teammanship between the FDA and the scientists and clinicians.
We have a question over here, ma'am.
Oh, yep.
Hi, I actually have 2 questions for you. Number 1, is your presentation available online?
I'm happy to do that. I can put it into a PDF. I must just make sure I've acknowledged everyone appropriately. I will do that because I did that verbally. So I'm happy to get it through public policy, or Carrie Monk will help organize that.
Great, that's very interesting. Number 2, your presentation triggered a thought, and for those of us who have been diagnosed with ALS, I'm sure we've heard from many friends, clients, family that you got to try this, there's another treatment, you got to go here, you got to travel to South America, you have to go to China. And we get flooded with so many different options, treatments, and I probably no different than most people, and done a few of those and really never thought much of them. But your presentation triggered something that happened to me a while back and had to do with electrical brain stimulation type of treatment. And I'm wondering if for all of those that are diagnosed and have reached out and have done different things, are you interested in any of the things that we have done, number 1? And number 2, how would we get that information back to you so that you could look at it and see if any of that makes sense? Because
those are very good thoughts, and I think that's the challenge with some of these alternative approaches, that there isn't good follow-through. So one of the things when people actually contact me and want to get a sense of whether they should or shouldn't do something, First of all, I'm not the clinician, so I don't speak as a clinician, but I would recommend that they speak with their neurologist, because if they decide any way to do it, it would be very important for the neurologist to be engaged with how they're measuring the different changes, what is being done, and so they can slot back into programs when they get back. It's an interesting thought of how to maybe, and it's something maybe we can talk with Rick and ALS Untangled, maybe there's a place to house that kind of information. The difficult thing to really make something of it is that none of them are done in very structured clinical trial ways. So, data is interesting, but if it's piecemeal, we will almost have to redo an experiment in the right way to understand it. But I think there's no harm to having a place to collect that information, to almost collect your experiences, positive or negative. And I think there might be, there are places like Patients Like Me and others, but if we can facilitate something like that, it's certainly worthy of thought. So thank you for raising that.
Thank you so much. I've enjoyed your talk very much. My question has to do with wondering if one of the challenges may be having to do with the pharmaceutical companies not wanting to fast-track drugs for the fear of lawsuits.
No, that's not at all the case. So in fact, there's an Orphan Disease Act which is put in place for rare diseases like ALS, which in fact is an incentive for companies to actually fast-track things. Industry has truthfully not been involved because of the huge hurdle. There haven't been clear targets, so it's quite a difficult disease area then to get into. The clinical trials are very long. Biogen would be probably the best example of some of the challenges that go into the trials. But I think they're better positioned. We're better positioned in the disease to test some ideas in a clever way. And as I said, the industry is changing. They are moving towards rare diseases because their pipelines are literally going off to generic compounds. They have many, many challenges. And rare diseases are actually an entry into maybe an opportunity to treat other disease areas with similar mechanisms, even Parkinson's and Alzheimer's. So no, I don't think companies are reluctant to go into it for any issues of that. I think they first need to really understand a clear path of how they can do this. It's a business model. You have to take the best guess of how to do it in the most cost-effective way. And that's, I think, where we play a huge role. What we've done in the past and what I do constantly, I talk to companies, we are willing to maybe test that compound in a model system or do some of the chemistries around it to improve it, or whatever it takes in a small way before maybe it becomes a priority in the portfolio at the company, where they have to, just like anyone is juggling a portfolio, they might have other things in their portfolio that is further along or looks more promising to be successful. The bottom line for them is success, clearly financial, but also to get it through the different stages. And I don't think they will embark unless they can see that path of success. And I think that's where we're actually being able to play a big role in bringing teams doing the early work to push that curve a little further to success. Yes?
A quick...
One moment. So I'll take a question here and then one here.
A quick thumbnail update.
I don't know where, I'm not seeing it though. Where is the question? Oh, thank you.
A quick thumbnail update of Brainstorm.
A thumbnail update on Brainstorm. I think that Merit's going to do a great job. Are you going to the clinical trial session?
Pardon?
Will you be at the clinical trial session at lunch?
Yes.
She's going to talk about Brainstorm. So let me, and then if your question hasn't been answered, I will address it there.
We've heard some recently about the understanding of ALS as a syndrome as opposed to a disease, sort of a conglomeration of different aspects that, of course, present in different ways. We can see that in this room. But then also in terms of treatment. And it sounds like when you're talking about the antisense treatment, we're talking about something much more mutation-specific. That notion of personalized treatment, would you be willing to comment on that?
Absolutely. It's a bit of a tricky one, right? Because you're making our population even smaller in terms of drug development. So that can be a tricky thing. But I think that the essence of a complex disease is the fact that, yes, you're right, there's such a variability. There's some with frontal temporal, some not. And so maybe just lumping all together may not be the effective way. Another way to think of it, and again, I'm not the clinical trials expert, but perhaps you do lump all together, but then in the analysis of the data, you sift it out more carefully. Because of the danger of being too strategically narrow, you might lose things because we are only using our best guess at what that narrow group is. But if you can cleverly make sure you've balanced out for genetics and for various things, that might also be helpful. Certainly in the case where we know the target is SOD, our first attempt will be to do SOD mutant families only. There is discussion, though, as to whether SOD is susceptible and sporadic, and that's still a very unknown. So then one might expand that, you know. So you're right in a way that personalized medicine is a direction to think about. It's a tricky one where you really obviously are going to have smaller populations. It also makes for very expensive medicine. It's an interesting story with cystic fibrosis for those who are not so familiar, and it's something I wonder how they're going to tackle. They did a fantastic, so cystic fibrosis is caused by a mutation. They are a great example of a philanthropy group that built public-private partnerships. Vertex, the company, has developed this treatment, which really is significantly cures the cystic fibrosis problem because it targets that gene. But it's phenomenally expensive. And now because they've really built exclusive opportunities, it's going to be interesting how we build into the generics and get cheap. So we have some interesting dynamics with very personalized medicine.
If you're going to tease it out later, that would require, though, that people know what their mutations are.
Well, I'm not saying that necessarily we would tease it out by mutation only. We would maybe do it by a panel of biomarkers. I mean, one example is perhaps there are those that have a particular inflammatory profile that is different to other people. And we say that because there's suddenly been an interest in a patient that responded very well on the neural stem trial, and maybe that's, you know, one thing to think about. And so maybe that will be a defining thing. We're working on targets where in some of the population it's elevated and others not, but it's not that that's the causative gene, it's just part of a mechanism. So it might not be just purely on genetics, and, you know, maybe the geneticists will have a different view, but, you know, we have to be careful to be too much chopping things, and then we might see things by serendipity. So that's just my personal view. There's no science behind that yet because we're still all struggling with that question.
Yes, thank you for the encouraging information. The other night, Wednesday night, I shook hands with a woman from North Carolina who told me that she was in a 6-month clinical trial that completely stopped the progression of her disease. And then, of course, the trial was over and the progression started again. I know that on the February 25th FDA hearing, one of the points that was made was, why can't patients who are positively affected by a drug, and she told me 27% of the patients in that trial had a slowing of the progression. Why can't we move those 27 to allow them to have that drug and continue to stop the progression? Do you think the FDA will positively consider that?
That's a very difficult one to answer, and I don't know if it's the Neuraltis trial you might be alluding to, the NP001. I don't know what that trial was. That's one of them.
It was that.
Okay. So there are a number of challenges there. One is I don't know, it sounds fabulous for the person who took that it was so effective. The data is still early, right, in a small group, so I don't know it to assess it as carefully as that to say it's that effective. But if it is, I see your issue and concern. The challenge is really also then for the company or the team that are developing it to have the funds to do it. And I think that is partly where they're at, as they're generating the funds to do the next phases. And it becomes a question of priorities. Is that the compound to invest in versus another one? But I don't know that there's a real block by the FDA for use of that, to be honest. I don't think FDA is the one that's blocking that. It's probably more with the investigators to try and do it. And I know that MP001 is a simple, I think it's sodium chloride. Anyway, it's something simple, but it's important that it's not just manufactured personally in the backyard, because there are toxic effects in the route it's administered. So it's not something you can just easily take on board. And that's the challenge. It's really getting backup and buy-in to get the funds to move it forward in some cases. And that's a question you can certainly ask Merit, and we can try and address again in the Q&As in the panel. Yes, I see someone waving.
Should I wait for the drug trial session, or can you tell us anything about the peptide called Hexa? It is shown to grow new synapses.
Oh, I'm sorry, I missed the name of the compound you said. Hexa.
Nahexa.
No. New Dexter, is that the one?
No. Dihexa. Dihexa.
So I don't have an answer for that, but I can look into it unless Dr. Bedlack knows Dihexa and is here. I don't see him now with all the lights. So let's see if Merit might know about that, and if not, we'll look into it for you. It's not familiar to me. It might come in another name which I would recognize, but we'll follow up. There was someone here who wanted to ask a question, and then over here. So at the back, please.
It was kind of an answer to the previous question. I was also in the NPO1 trial from Neuraltis. And last year at this meeting with Congress, one of my congressmen actually wrote a letter to the FDA, and the FDA gave him a pretty clear outline on how to get compassionate use for the NPO1. But then when it got back to Neuraltis in California, they pretty much said that they're a research company and they contract out on making the medication, and that they weren't capable of making the medicine in the quantity that we would need for compassionate use.
Thank you. That helps clarify that very clearly. Thank you.
Yes, is the research on Rayazole, is it completely dead? It is our only FDA-approved treatment, and it's been around for about 30 years, but you never hear of a new and improved version of the drug.
We are funding a group actually in the UK who are just doing exactly that. What is difficult about the drug, and we have funded in the past several studies to try and really highlight it, is it's not clear what the mechanism of action is. We think it works on the glutamate process, but it's not really very well defined how exactly it's working. So that does make it difficult to improve a structure where you don't have a very well-defined single target. It's probably because it's a dirty drug, so to speak, has multiple targets, that it has some effect. And it's been quite difficult to tease all of that out, to be honest. But there is a group, a chemistry group in the UK who is working on trying to design some different molecules with a similar backbone to see if they are effective as well. Thank you. It certainly is. It's something that keeps on coming up, and we wonder why we don't invest more. And I think partly it's been a limitation of how to go about to do that. I think it's time to break. I haven't checked the time again, but I hope you have a chance for a coffee. And thank you for your good questions and all your interest and participation. Thank you.